Thanks very much. So firstly, thanks to the organisers of the long programme. I've been here for the last nearly 4 weeks, and it's been great to be in this environment, talking to a lot of interesting people and getting space to think. And thanks to the organisers of this workshop for adding me to the schedule as well. So this is the talk I said I would give, and then I realised I had a 50-minute slot, so I needed to fill some content. So I smooshed 2 talks together. I was live editing the slides 5 minutes ago. There's probably going to be a disaster in there somewhere, so please forgive me if there is. And it probably means that I'll also run over some things too quickly. But I always like the opportunity to try and make a point about how we should think about things. So I'm going to try and run through some prior work pretty quickly as part of making the general point, and then I'll get to the actual topic of the talk. Now, in my experience, this audience is very keen on asking questions during the talks. So I would ask you maybe in the first half, when I'm presenting the historical stuff very quickly, if it's not clear, please don't slow me down, otherwise we'll never get to this part. And I'm happy to take questions afterwards, but that stuff is kind of quite old now, and I'd like to show you some more recent results that I've been working on. So I'd like to start by acknowledging Giovanni Reina. Most of the work I'm going to show today has been done with him. He was in my lab for over 5 years, and he's now a research group leader in Konstanz, collaborating with Ian and others over there. And as I said, this is a talk in 2 parts. So in part 1, which I'm going to go over fairly quickly, I'm going to talk about how we can think about modeling collective animal behavior and just analyzing it in general. And in this, I'm drawing inspiration from Nico Tinbergen, pictured here, who, along with Konrad Lorenz, won the Nobel Prize for basically inventing the field of ethology, or animal behavior. Now, Tinbergen posed 4 questions for thinking about animal behavior. And his 4 questions, actually, he was kind of elucidating or extending ideas proposed by Ernst Mayr as well. And between the 2 of them, you can come up with this kind of categorization of behaviors. And pretty much anything in biology. The first is the proximate view. So that's the mechanism, the cause. How does something work? And then there's also the ultimate view, which is the why question. Why has a behavior evolved? Not how does it work, but why does it actually exist and function in the way it does at all? And then you can take a dynamic view of things, which would be an evolutionary perspective or a kind of developmental perspective within an individual organism's lifetime, or you can take a static perspective. And when you fill in the blanks here, what you end up with are standard questions that animal behaviorists, behavioral ecologists answer. Proximate dynamic is behavioral development. How does a behavior develop? But it could be, how does an organism develop? How does an organ develop, for example, during the lifetime of an individual? But I'm focusing here on the behavior. The proximate static view is what's the mechanism. The ultimate dynamic view is the evolution of a behavior over time. So over evolutionary time, is it progressively refined? And then finally, the ultimate static view is behavioral function. Now, anyone who knows me at all well will know that I'm highly fixated on the ultimate explanations. I really want to know the whys. Why is something the way it is? And not so much on the mechanism, but there's actually a huge amount of interest, I think, in melding these 2 views. And some of the work I try and do does try and combine the proximate with the ultimate view. So if we can figure out the mechanism, but also the why of the mechanism, that's really a complete picture. And we've seen some really nice examples of that earlier in this workshop and in previous workshops as well. And Ian's talk from earlier in the week springs to mind, for example, looking at function and mechanism together in animal navigation. But so what Giovanni and I did was adapt this then to collective animal behavior, and we said that there are 3 questions for collective animal behavior. If you're ever trying to apply any of these lenses to look at a particular behavior, these are some common pitfalls and some questions you have to answer thoroughly before you try and do so. First question is what quantity should be optimized? So if you're going to look at optimality theory, which is the why question, not the how question, then you have to figure out what actually natural selection should be optimizing. You might get it wrong. You have to figure out what behavioral mechanism should be used. Again, you might make a mistake there and figure out that just assume an incorrect behavioral mechanism, play it through, and then come up with some counterintuitive result. Whereas, if you identified the correct behavioral mechanism from the start, you wouldn't have necessarily drawn the same conclusions. Now, these are both questions that could actually be asked in any aspect of animal behavior using Tim Bergen's 4 questions. But I think there's an interesting third question here, which is particular to collective intelligence, collective animal behavior, the subject of this workshop, which is, at what level should behavior be optimized? Because here we actually see, in collective animal behavior, we see actually things that on the surface look quite similar in that they're composed of multiple units interacting to generate some aggregate behavior, but actually the nature, the evolutionary history of those systems can be very, very different. So to give you an example, and we've seen, I think this a couple of times already, the starling murmuration. Now, this is a group of unrelated individuals that exhibit highly coordinated behavior. And there are various possible functional explanations for that. Maybe it's predator avoidance. I'm not an expert in that, but what I would say is there's no way that the behavior of the individuals can be optimized in order to, or can be shaped in order to optimize some group level behavior of the flock, unless some very particular things hold. But here, this is a population of unrelated individuals, so somehow we have to explain the complex aggregate behavior by selection acting on individuals, the individual behavior within the concept, the social context. Now that's very different to the kind of systems I normally look at, which are social insects, where you can effectively treat the whole social insect colony as a superorganism because they're all highly related to each other and their evolutionary history has shaped the behaviors of each member of the society. We've seen that in, I think, army ants earlier, and I'm going to show you some honeybees and other ant species. Every individual can also almost be thought of as like a cell in a body, in a superorganismal body, each adapted by natural selection to fulfill their role within the context of the whole performance of the colony. And some other interesting things come up from answering this question as well. So Ian earlier posed a question of Richard in his talk on confidence weighting. So this caused me to put in this, a plug just for this old result. So again, if you take the confidence weighting question and you look how confidence signaling or confidence weighting of opinions within a group based on information quality or other factors might be accounted for, actually you end up with different prescriptions for how opinions should be combined according to whether it's a group of unrelated individuals, in which case the individual, the signaling individuals may actually be deceptive, for example, or it's a society of highly related individuals like an insect colony, where there's this co-adaptation, co-evolution of their behavior, and actually the signaling is much more likely to be trustworthy and honest. So I think this is a kind of productive area, an interesting area for these 4 questions for collective animal behavior. But what I'm going to do is take you through 2 examples under my favorite lens on the left, the ultimate view, and I'm going to start with behavioral function. This is a slide that I should have taken out or populated, but I'll briefly talk to it. So if we're going to look at function, we're going to use optimization tools to try and analyze behavior. We're going to import tools from economics, statistical decision theory, et cetera, et cetera. And what I particularly like is anywhere where we can formulate a problem and we actually have a mathematically provably optimal solution to the problem. I'm really happy because then I can look for, is there a biological instantiation of that solution? And how can we interpret the behavior we see through that lens? So the examples I'm going to start off, the example I'm going to run through very, very quickly is behavioral function in house hunting social insects. This is a honeybee swarm. So periodically the honeybee colony reproduces or is forced to relocate. They're now highly exposed, in the case of the honeybees, to the elements. They're unable to forage. All of the foragers here are completely engorged with honey, so they have limited energy reserves. They have formed a mantle around the queen to protect her and keep her warm, and they're exposed to the weather, unable to forage. They basically need to find a new nest site very quickly. So this is a choice problem, and it's the kind of choice problem that neuroscientists have looked at and psychologists have looked at extensively. At its simplest, it could be down to a binary choice problem. Should I choose hollow trunk A or hollow trunk B for founding the new colony site? Now, neuroscientists have looked at this extensively in these kind of psychophysical tasks like random dot fields. This is where I'm going to start going very rapidly because this is old stuff, and I don't want to get bogged down in explaining it. But in collaboration with some very inspirational colleagues like Raphael Bogac, who was with Phil Holmes and Jonathan Cohen at Princeton for his postdoc, we looked at how neuroscientists had modeled this choice behavior and how I was modeling the choice behavior of the insect colonies, and we found some really interesting correspondences. And the optimality theory imported by the neuroscientists was sequential decision theory or drift diffusion models, which I think we've again seen used in some of the talks earlier. It's basically, this gives you a provably optimal compromise between the speed and the accuracy of the decision making, but it also has a very nice kind of physical analogy, which is a process of integrating a Brownian motion noise process with a constant drift towards a decision boundary. And it all kind of falls out nicely. You can represent the signal, the noise, reaction time, distributions, all kinds of stuff, all falls out really nicely, so it's very attractive. So the neuroscientists have hence done a lot of this kind of stuff, applied this theory extensively to choice behavior, whether it's simple right-wrong choices or if it's even value-based choices, such as choose which of these two things you like best at lunch, at breakfast. Now you probably do have a preference, you probably have an instinctive choice for one or the other. Doesn't really matter which, but as long as you can integrate your evidence and make a choice, then get the reward, then you'll be happy. But the interesting question in these kind of choices, unlike the random dot task where the monkey is rewarded according to whether it got the right answer or not, it gets a juice reward if it correctly classifies the motion stimulus, and it doesn't get a reward otherwise. Here, regardless of whether you chose the thing you like most or not, you nevertheless get what you chose, right? You might not like a pain au chocolat, you might prefer a croissant, but still, you choose the croissant by mistake, you still have the croissant to eat. You get that reward. This is called value-based decision-making. And what's interesting, gives an interesting departure from the psychophysics we were looking at with the random dots. What happens when you have a choice like this, where there's only croissants? Now there's no difference to integrate in your choice. You should just choose one at random. They're basically functionally equivalent, right? But the drift diffusion model, crucially, the drift term there will be zero. The signal in the decision process is zero because there's no difference in the qualities you're considering to integrate. And the drift diffusion model in such a case will integrate noise until it reaches a threshold. In other words, you only reach a decision by integrating sufficient noise and taking sufficiently long over it when there was actually no choice to make in the first place. So this led us to the bees. The bees we presented with a very similar situation, which was presenting them with identical artificial nest boxes. We found a nice motif, which is a mutually inhibitory motif, which is very similar to what's proposed in neural decision-making circuits in visual cortex and other brain regions involved in choice. And it had a bunch of nice decision dynamics that very elegantly solves the problem of how do you deal with situations where sometimes there's a real choice to make, but sometimes there's actually no choice to make, and you should either maybe adaptively maintain a decision deadlock or adaptively break a decision deadlock based on the qualities of the options you're considering. So for example, if you really loathe croissants, maybe you shouldn't choose a croissant at all if that's all that's available. You should wait and see if someone's about to bring out in a few minutes the tray of things that you really do like. So it's kind of quite a complex decision circuit. We were very happy to find it, and we thought it maybe spoke to the neuroscience a bit, because we felt the predictions being made for value-based choices from the neuroscience theory were probably incorrect because they'd identified the wrong optimization criteria, that you should trade off speed versus accuracy in decisions. And we proposed instead you should trade off speed versus value of decisions, which is exactly what this circuit does. But this is just a dynamical system description. It's a descriptive model. Yeah? It's a mechanistic model that explains some aspects of choice and how choice could be made. We could say adaptively, but we don't really have any formal reason to say that it's the best solution. So how do we know this strategy is optimal? Well, here, other colleagues from neuroscience actually imported stochastic dynamic programming, which is an optimization tool most of you are probably familiar with. It actually has a long history of application in behavioral ecology. Maybe, I'm not sure so much in neuroscience. So they basically set up the dynamic program for decision making. So they solved the Bellman equation for the decisions where decision makers are rewarded by the value of the option they choose. So the Bellman equation then looks like this, that the value of deciding, given some estimates of the values of options you're considering at a particular point in time, is basically the value of sampling you get in the future with a discount, and minus the cost of sampling you pay for taking longer over the decision. And what they found was actually the optimal solution for value-based decisions was apparently drift diffusion models with decision boundaries that collapse over time. Or in other words, that agents should optimally solve the speed-accuracy trade-off. And in particular, this doesn't kind of fit with any of the behavioral work that's been done recently in these deadlock cases, where we actually, in particular, find some really interesting relations between reaction time and values of options being presented. And I'm going to show you some data on that very shortly. But so how can we explain this? It looks like they applied optimality theory and they came out with a prediction that things should be the way that they don't actually appear to be in real life, that real agents don't seem to conform to, and that also doesn't actually seem to intuitively align with how we feel the problem should be solved optimally. Well, the devil was in the detail here, so we took their dynamic program and adapted it. And the main change, in fact, the only change to make, was in the way the sampling cost was accounted for. In the original dynamic program, time had a linear cost. So your time you spend now is the same as the time you spend, 10 seconds in the future. But that's very non-biological. It's also not really how Bellman equations are normally formulated. Normally, instead, you have a geometric cost of time, right? So you just scale future rewards by how far they are into the future with some discount parameter gamma. And here we were actually able to recover the kind of empirical stuff we were seeing with a variety of biological agents and come up with strategies that optimize the speed-value trade-off. So, this was actually my son under lockdown, along with 100 other participants doing a big online experiment, which was a pure, unrewarded, value-based choice experiment. And you can see here, it was actually just psychophysics as well. It was discriminate the brightest of three stimuli, and you can see the brighter the stimuli got, the quicker the reaction times got. With Audrey Dussator, we worked on slime mold. Simon Garnier presented on slime mold at one of the earlier workshops in this long program. I recommend going back and reviewing his talk if you haven't seen it. Again, we set up the same paradigm for the slime mold, and we saw exactly the same effect, that the concentration of the stimuli, in this case nutrient patches, again, negatively correlated with reaction times. And that correlates with the optimal strategy we found from the dynamic program. So the kind of lesson is, and people have been looking at speed accuracy trade-offs in neuroscience and psychology for a very long time, and actually even that theory got imported into collective behavior, and I worked on it myself in house hunting ants. But I would argue that actually there's just a fundamental misconception there in actually what the organisms should be maximizing and how they should behave as a result. Now, that can be quite hard to shift the inertia of a particular field once everyone's very committed to a very nice theoretical paradigm, but still, I think if we're really trying to understand organismal behavior, we need to actually answer these questions correctly when we're applying optimality theory in order to correctly interpret what we're actually seeing in the real world. All right, so that's my kind of evangelical portion of the talk, where I just try and persuade the audience of the value of thinking about these distinctions, Tinbergen's 4 questions, the value of applying that to collective animal behavior, and the importance of getting the optimization criterion right, in particular, whenever you're trying to do this kind of work. So in part 2, I'm actually going to give the talk that I submitted, which is to just give you another example of this, but much more specifically looking at noise suppression in collective foraging.
Jason, can I ask a question?
Yeah, you can now. Thank you. Thanks for waiting.
So this is really cool. I just wanted to make sure I understand. The proposal is to be speed-value trade-off as opposed to speed-accuracy trade-off. And there are some conditions in which accuracy and value would correlate very highly, because supposedly the accuracy is defined in a way that is rewarding in a task.
Yeah.
So are there cases that you tried where the two are either orthogonal or not highly correlated?
And we actually saw this earlier with Ahmad's talk, that so if you can learn the values of the options and they're static, then you can, optimizing the speed accuracy trade-off can be equivalent to optimizing the speed value. But you have to, if the world's going to fluctuate at all, like if you can't predict what the values of the options will be and you have to actually gain information about them during the decision-making process, then what I just showed, the different optimal policy is the one that optimizes the speed value trade-off. It's kind of like doing a bandit problem where you're rewarded during the choice process in some way. No, that's the wrong way around. A banded problem is like the ongoing kind of version of this decision problem, where you accrue reward during the process, whereas here you have to sample and then eventually choose and you receive the total reward from the option chosen.
Latent state accuracy could be defined as just finding the highest value option, which is the problem in reinforcement learning, for instance, which is why I just say
yeah, absolutely. But the question for a biological organism is, outside of the lab, there's no one externally saying, well done, you chose the best option, you get the reward. They get what they choose. Okay, so I'm going to talk about noise suppression and collective foraging as another example of using this kind of framework. And again, the importance or the interest of looking at what's the correct optimization criterion. And if you haven't been down to the Hammer Museum during your stay yet, then please go and check out these amazing bee artworks, which are currently on display, and even a live bee artwork exhibit. If you don't want to go there, you can actually stream it online from the Hammer Museum website as well. At any point, you could even do so now. But it's well worth a visit in person to go and see what's happening. So here I'm moving from house hunting to foraging, but actually the model we're going to use will be pretty similar. And we're going to look at behavioral function again, but then we'll actually be able to, with the same model, start looking at behavioral evolution, which is pretty rare, I think. Okay, so let me start with a quick primer for signaling mechanisms in social insect colonies. Now, positive feedback signals in social insect colonies are probably widely known to even lay members of the audience who never work with insects. So positive feedback in ants, for example, I worked on Temnothorax ants early on. This is an example of a tandem run, which is a one-to-one positive feedback process where the lead ant has information about where to go, and it physically leads the follower ant there such that that ant can then independently repeat that journey and turn teach others and so on and so forth. So that's a positive feedback signal. Obviously for pheromone-based ant species, you have mass recruitment, pheromones, the army ant trails, all this kind of stuff. So positive feedback is well understood. In the honeybees, the waggle dance, similarly well understood and well recognized as a positive feedback signal. Now it's a kind of mechano-sensory process of information transfer where a bee knows a vector to a particular point in space, could be a forage patch, could be a nest site, and the bees following it here and maintaining antennal contact can actually decode that vector into a vector to fly in the real world. The vector in the hive is encoded relative to gravity, but it actually is decoded relative to the sun, which is really cool as well. But only comparatively recently discovered really in social insects are negative feedback signals. So here are Pharaoh's ants, and this was work that Elva Robinson did with Francis Ratnieks quite a while ago. So monomorium, and you can see here a smoke-plated glass sheet, and you can see the pheromone trails. Sorry, you can see the trails that have been followed historically because they've been eroded by the ant's feet. And what this species is able to do is actually, if it goes down a particular branch of a trail that has previously been rewarded because there's pheromone deposited there, and a number of ants have gone that way, and it finds no reward, no food reward, it comes back and it actually lays a repellent, no-entry pheromone to stop others wasting their time going down there. And that was pretty much, I believe that was one of the, if not the first demonstrations of negative pheromones, which had been theoretically predicted. In honeybees, you have the same kind of principle, but it's just differently implemented. So this is a waggle dancing bee, and this is work that we did with Kirk Vischer and Tom Seeley. This bee is, the yellow and blue marked bee has been to a nest box where it's been marked with paint pens, and it's advertising using that waggle dance the direction to a particular nest site. But you'll see is actually the bees that are following it have been marked with a different paint pen color, which means they have information about a different nest site. And actually, if you can hear the audio well enough, and I hope you can, every now and then you'll hear a very short, high frequency, high energy burst of noise, yeah? It's coming there. Yeah? And when that happens, you'll actually see one of the followers will lunge towards the dancing bee and headbutt it and kind of transfer that energy into its gaster. Now, that's very disruptive. So it's called the stop signal in honeybees. And it tends to shut down the waggle dance. Dancing bee is discouraged, as you might be as well. So these negative feedback signals have all been observed previously and identified in various contexts and proposed as mechanisms to deal with fundamentally foraging sources not being ideal. And here are the examples. The pharaoh's ants example I mentioned was, it's a time varying environment. So you go down a branch and the food is no longer there. So you should just shut that branch down, shut that pheromone trail down. Similarly, I think this is bees. If a forage site is very crowded, so there's a low efficiency, a low kind of nutritional uptake efficiency at that site, you can have a negative feedback signal to slow down recruitment to that site. If you simulate predator attacks on bees, James Nye was able to show that the stop signal will also be delivered to any bee advertising a particular forage patch where they've previously experienced a simulated predator attack, because they have some natural predators that wait for them at forage patches and attack them. But in this work, we're going to propose an alternative function for negative feedback signals, which isn't about any of these whatsoever, but is instead about reducing the variance of the colony performance. So again, this is an example of, think back to the framework, the Timberger's 4 questions. What quantity should be optimized? And in behavioral ecology, there's a long history of thinking, even especially about foraging insects, what's the correct optimization criterion to use in explaining the behavior of an individual forager, never mind a collective of foragers. So a long tradition of study. So the model I'm showing you here is actually exactly the same model that we derived for the house hunting honeybees, the swarming honeybees, but it's just been adapted to foraging. And in fact, it wasn't even adapted to foraging first by us. It was adapted by Zach Kilpatrick and colleagues, I believe it was. And so this is the first time you're going to see the model. So I'm going to work through it so you get an intuition of how it works. But it's a very simple kind of compartment model or state transition model. So individual bees go out and find forage patches independently. And they can also spontaneously abandon them. So, the number of bees aware of a particular forage patch and committed to it can change through individual non-social action, independent discovery, and independent abandonment. But then they can also recruit others through positive feedback, like the waggle dance in the bees or the pheromone trails in ants. But they can also inhibit other bees, and they can condition that inhibition on, their relative information, what options they're each interested in, through stop signals in bees, negative pheromones in ants, and so on and so forth. So it's a very simple model that you can describe as a chemical reaction model, where you have different species, different chemical species, insects committed to food sources, insects that are uncommitted. You can describe the chemical reaction transitions as being transitions that happen at particular rates. So discovery, a bee discovers a site at a rate gamma associated with that site and abandons with a different rate. But then 2 bees can interact where they have different information, and one of them, as a result, will change the information it holds. That can happen at some other rate. And all of these rates can be conditioned on the quality of the different options under consideration, all of this kind of stuff to make a pretty rich system. And then inhibition is kind of the flip process. 2 bees, in this case, 2 bees that have the same information, one will inhibit the other and flip them back to the uncommitted state. Now, all of these colors, the blues are all one particular food source, but you can just scale that up to as many food sources as you want to get a broader, a larger set of reactions. And then put some qualities in there so that the rates can be conditioned on the qualities. Okay. Any questions on that before I move on, just on the construction of the model? No?
Just on the inhibition, so does that happen in this case, where they have the same information and the one is inhibiting the other, rather than the case the video seemed to be?
So it depends on the behavioural context. So for foraging, yeah, absolutely. This has already been documented that a bee will observe a waggle dance, say, yeah, you're going where I know is bad because of the predator attack, for example, and I'll shut down your dance so no other bees go there and risk getting eaten. In the house hunting case, we found the opposite pattern, which was the cross inhibition, because that was actually what let them converge on a single option and break these deadlock dynamics, break these decision deadlocks. So yeah, in the two cases, you actually have different patterns of interaction.
And a single bee can know if its signal or where it has been is better than where another bee has been, or does a bee go to both places?
Yeah, well, that's a bit of a debate in the literature, at least in ants. The models don't require them to, or assume that they do, in fact. They just, by having the individual rates conditioned on the individual estimate of the quality of a site, the dynamics overall will be such that it's like the colony is effectively making a comparison, even though no individual needs to. Ida?
So the beta ij is always dyadic, or is it sometimes in an average of bees around me or something?
Yeah. For the models, it's always dyadic, but you can have lots of these. Depending on the concentrations, the frequencies of the different bees of the different types, you can actually have quite a lot of these happening. When there's good intermediate frequencies of the committed and the uncommitted, or what have you.
So do they then sample from different populations, or how do they do that?
Well, so this is a chemical reaction model, so everything's basically like we assume it's a well-mixed system. So at the end, in the infinite population limit, you're going to get a differential equation system out that describes the behavior, the time evolution of the system. But that won't be the interesting part. Or it will be, but it'll be not the full story. And we won't even assume anything about how quality is represented, what it really corresponds with. All we'll assume is that if there's a Q1 and there's a Q2, then there's some preferred distribution across the different patches, because there's a lot of different foraging theory, right? There's ideal free distribution, probability matching, all kinds of different ways in which you might say the colony should distribute its resources. We don't care, we just represent a target distribution with these two parameters, Q1 and Q2, without saying how it's arrived at, what it actually represents. As long as it can be sensed by individuals, that's all we care about. And so we're going to look at I shouldn't have skipped back there. We're going to look at the role of inhibition here. Crikey. That was a good use of my time. Apologize for that. So we're going to look at the role of the inhibition process. We're going to look at the model without negative feedback. So we'll just completely remove that process. And we'll compensate all the other rates so that the foraging efficiency of the colony is not compromised. And then we'll look at a model. There's some mathematical details. Don't worry about that too much. And then we'll look at a model with negative feedback. So as I say, we will make these, there's clearly another process at play here which changes the distribution of foragers over time. So we basically make this a fair comparison as far as possible, so that neither foraging colony has the advantage over the other because it does or does not have this inhibitory process. So we're only looking at the impact on the dynamics for the performance. Everything else is corrected for. So we get rid of the confounds. And we had lots of different food sources. And we can do the deterministic analysis of these systems. So we can derive, as I said, the differential equations that describe the time evolutions of the populations of foragers. And you can see that both, so what we have here are basically these are the ideal target distributions in the dashed lines. And you can see that in both cases, the systems can converge to the preferred distribution over the forage sites, right? So it's going to be, let's say there's 3 flower patches. It's going to be some proportion of your bees here, and some here, according to whatever the cues are representing. Okay, so both with and without negative feedback, we're able to converge to the desired distribution of our resources over, for resource collection, our bees over the available resource patches. But when you actually bring in stochastic effects due to a small population size, the story looks very different. On the left, so here we're using the stochastic simulation algorithm, so we actually explicitly do probabilistically correct simulations of repeated evolutions of the population trajectories. So it's effectively, we're sampling from the master equation for a fixed system size. And you can see on the left, a very high variability in the performance of the foraging colonies. That's the one without negative feedback. Whereas on the right, you can see that the variance in performance is much reduced in comparison. And as you'd expect that is indeed significant, highly significant. So the kind of errors around the target distributions are very high without negative social feedback and really very low with negative social feedback. So there's a variance reduction. And you can do a sensitivity analysis and see that, in pretty much the entire parameter space this holds. So it's not just a special case of the parameters. It's a very broad finding. And more of the same. So here, just changing the recruitment. So the previous one was changing the recruitment, strengthening the system size. And as you'd expect, the effects are most marked when the system size is small and reduced as system size increases, because then you're getting closer and closer to the expected behavior of the infinite population. And then as you vary the recruitment strength and the food patch quality, again, this is still a consistent difference. So the variance is very, very low across all parameterizations when you add negative social feedback in. But there's an interesting thing when you look at the speed of the convergence of the system. So eyeballing it, it looks like actually they converge to the target distribution kind of over similar timescales. Maybe it's like an order of magnitude difference here. But there's actually quite a lot of difference. The model without the negative feedback, so here the model without the negative feedback looks quicker. But that was for symmetric initial conditions. When you change to different initial conditions, asymmetric ones, then you actually find that the model without negative feedback takes far longer to converge than the one with negative feedback. So negative feedback is again proving useful here as well. And then what you actually end up with, if you play with this further, is a speed robustness trade-off. When the strength of the negative feedback signal within the system regulates this trade-off. So in the red curve you can see is convergence time with the units on the left, and the green curve. Apologies if you're red-green colorblind. I just realised. This curve is the error around, or the variance around the target distribution, and you can see basically by changing the strength of the cross inhibition, the negative feedback. You have to trade off between the speed of convergence to the target distribution and the error around that target distribution. So, to summarize that, negative feedback, we propose, could actually be a variance reduction mechanism for social insect colonies. Now, we already knew that it was good for breaking symmetry in collective decisions, for example. That was the swarming work with the bees, or adapting to time-varying environments with the pharaohs ants and the stop pheromone. But here we're finding that there's a really interesting potential role in variance reduction. And this is actually similar to what you find in gene networks. So in gene regulatory networks, you can again find the same motif, that negative feedback helps stabilize those networks. And also in electronics for reduction of noise in amplifiers. It also explains some empirical observations that have previously been unexplained, such as, for example, even in static conditions, honeybees have been observed always delivering a small number of stop signals to foragers that have visited the same patch they've just visited. And that was observed quite some time ago, but it doesn't really have a functional explanation yet. This is a candidate functional explanation for that. Okay, well, let me just round this out then by just returning to the 4 questions that we started the talk with. And now we're in a position to actually potentially even come up with a suggested, proposed answer for a behavioral evolution question. So an evolutionary answer on a dynamic question. So how could the evolution of this mechanism have taken place? And it's pretty simple, really. So the proposal is this. In the beginning, you have pretty much solitary insects, foragers, no feedback, low efficiency, because they have to discover all their own food sources. And if the food source, food provision is variable, they're going to have to go out repeatedly. They're never going to get the benefits of information from other foragers that have found other food sources. So it's low efficiency. And they can't also recruit, they're not colonially living, or if they are, they're still not communicating in a way that enables them to recruit others to go and exploit a food source they themselves have discovered. But then the next step in the refinement of the collective foraging system could be adding the positive feedback component. So now you get the efficiency gains because when new food sources are discovered by others, you're able to, you also get access to that information, and you can collectively build up a population of foragers to exploit the resource more efficiently. Especially important if resources are transient, if they're ephemeral, if they're only going to be there for a while and need to be exploited quickly before a larger animal comes and takes them or something. But that solution is noisy, as we saw. There's high variance in the performance of that system. So the final step in the elucidation, the refinement of the system, is to add the negative feedback signal, where you now reduce the variance of the colony level performance, and it also becomes more adaptive with this trade-off between the speed of convergence and the variance of the colony's performance. And that's really it. So those of you who are on the long program and came to my tutorial last week may recognize that this kind of system can be modeled with the tool that I gave a tutorial on last week. So actually this analysis can all be reproduced in this nice tool that Giovanni and I co-developed, which lets you do a lot of collective behavior modeling, including very advanced statistical mechanics analyses without actually typing any equations in yourself. So that can be nice. It also is kind of a complementary approach to the tool that Vishu presented earlier in the week, where he comes to the same destination, master equations and Fokker-Planck equations, but from time series data, so you can actually derive. So apart from that, just thank collaborators from my lab and elsewhere, and funders. And if you're interested in the papers, then please just scan that code and you can download them. And I will stop there, and thank you very much.